Animals used to play a key role in human primitive cultures, and they were accorded special attention. They were often agents of fate or carriers of messages. 32,000 years ago, in the caves of Chauvet, human beings painted rhinos, lions, horses, bears. Why didn't they paint other men or women, or fire, or water, or weapons? All those things were certainly very familiar to them and also essential for their daily survival. Why animals then? Maybe because their life depended mostly on animals or because they were their competitors. However, those paintings unveil a close and selective attention for animals, revealing a deeper interest. Animals have something mysterious. They are extremely different from us. For instance, it is impossible to say what goes through the head of an animal. They are like aliens, undescribable. However, at the same time, they are very similar to us, sharing our common condition of being alive. I would like to link up with what has just been said to summarize the reason of our presence here as follows. We observe life without forgetting that at the same time, we also host life just as any other living creature does. The title chosen for this journey together, Being Alive, intentionally refers to the idea that life is not only something that we can observe and scientifically investigate, but also it is something that we experience directly. It is a condition that we share with all the other living beings. However, as humans, we are certainly privileged. On the one hand, thanks to scientific research on living systems, we keep discovering more and more surprising and interesting scenarios that we would really like to show you as far as possible during this journey. On the other, each one of these scenarios brings us back to some sort of original condition, and by analogy and almost empathically, we recognize this very same condition also in all the other living creatures. It is all about being living subjects, in other words, being alive. This condition implies the fact of living in a world that is full of meaning that we give and receive at the same time, as any other living creature does. Each living creature in its own way embodies a perspective on and in the world, a world that is inevitably made of other living creatures, as shown by the animals depicted in the cave paintings. But it is also overflowing with life, as portrayed by this painting by Jan Bruegel, The Allegory of Water. And keeping this in mind will help us appreciate, in a maybe new way, the hidden and intricate landscapes we're going to discover. Having just mentioned astounding scenarios disclosed by scientific research on living systems, I would like to start by introducing to you two major features constituting hallmarks of all living organisms. The first ones are the biological building blocks making up any kind of organism, the cells. This great variety of shapes and organizational complexity already tells us a lot about the plasticity and complexity of living systems. The second ones are two essential categories of biological macromolecules: proteins and nucleic acids. They are both polymers. In other words, they're very long molecules made of repeated, simple units called monomers. A key feature of these molecules, namely the RNA and protein ones, about which we're going to elaborate more later on, is their ability to spatially fold to form differently shaped 3D globules, as you can see in this picture. Another peculiar feature of these macromolecules is that they are not blindly assembled. On the contrary, they result from specific chemical processes of ordered assembly of monomers based on instructions that we're going to see in more details later. However, I really want to emphasize the absence of such processes in non-living nature, because this is a unique hallmark that can be seen and experienced because it affects only living organisms. Special attention should be paid to proteins because they are involved in any vital structure or process. Every organism has dozens of thousands of different types of proteins. Each of them is the result of the combination of monomers, amino acids, resulting from a set of 20 different types that, just like letters, can be combined in countless ways, and they can form large, unique 3D globules involved in crucial processes, and they can form 3D objects and really be part of key processes. In the video, you can see a folded protein made of amino acids which behave just like alphabet letters. And actually, the letters you see here refer to abbreviations indicating the name of each single amino acid. This folded shape makes up a globular structure including other folds and pockets and crannies. But none of them is accidental. Not at all. Everything is strictly related to the specific task fulfilled by the macromolecule, which is not conceivable, not even explainable, without considering as a part of a longer context, including many other proteins. This is exactly the typical living system scenario that we are going to gradually discover together. One of the usually most surprising features of living systems is their ability to keep their own identity over time, in spite of the almost continuous and deep change of their material components. Almost all the atoms making up my body right now are different from those that were in place just 1 year ago. Even if my atoms have been almost completely replaced, I am still myself. Each living system is not purely and strictly identified by the matter it is made with. Instead, it is characterized by an extraordinarily complex process, according to which matter and energy are, so to speak, harnessed in a highly organized dynamic system whose persistence does not depend on the persistence of its single material parts, but on the perpetuation of the whole process itself as a whole. In order to better understand this, we can think about the video images showing a composite structure made of constant in and out flow of building blocks. And the structure keeps existing within this flow, changing its shape accordingly. So the structure can grow, it can shrink, or simply adopt a different shape. This illustrates quite well a key feature of living systems that is not always straightforward, because we tend to perceive living entities as something static and fixed. On the contrary, they're constantly changing, as wonderfully shown by familiar pictures we do not wonder enough about, like those illustrating the growth and development of living beings. Another extreme example of this dynamic behavior is the embryonic development seen in the metamorphosis of tadpoles. Once again, I really want to underscore how living beings are constantly changing, even when they seem to be static. Actually, the processes allowing an adult organism to keep its shape are exactly of the same nature and type of those responsible for its growth and differentiation. Shape and transformation are inevitable concepts when talking about living systems. The Greek word metabole, meaning change, mutation, is at the origin of another key life-bound term, metabolism. Metabolism refers to the process used by a multicellular organism, like me, to literally transform and integrate parts of the outside world through an incredible set of interdependent chemical processes occurring repeatedly in each one of its billions of cells. However, it is paramount to understand that the metabolic process cannot be simply compared to the process allowing a machine to get the fuel it needs to function. There is much more than that. As a matter of fact, nutrition provides any organism not only with the energy it needs to work, but also, more radically, with the very same atom-made matter that makes up their physical bodies. And this marks a clear difference between living entities and machines. Living organisms self-build themselves. But how is it possible? This is clearly something extremely complex, but in order to get an idea of how it works, we can have a look at one of the most important metabolic pathways, the glycolysis, which consists in the conversion of glucose, a sugar, into pyruvic acid or pyruvate. The molecule is broken into two molecules, releasing energy usable for other processes. The pyruvic acid can be further degraded to release further energy. Glycolysis might seem to be a simple process, actually, even though it looks simple, and even though this diagram is not clearly detailed, it is a complex sequence of 10 reactions, allowing the conversion from glucose to pyruvate, as you can see on the right-hand side. But this is not all. In fact, pyruvate can be further degraded into acetate, and then further degraded again, as shown in this cycle, to release CO2, and this pathway leads to energy release. But importantly, pyruvate can have other fates. In particular, as shown here, it can also be converted into alanine. Why is this important? Because alanine is an amino acid, one of those monomers that can be used to make up proteins. Therefore, the original glucose atoms resulting from food intake can become proteins and so become part of our flesh or muscles. Now, we may ask, how many molecules and reactions does the metabolism include? Well, in order to understand this, the answer can be found in this extremely candid general overview of the metabolic network. It is a huge, intricate maze of thousands of interconnected reactions. The products of a reaction become the reagents of another reaction. Metabolism is made of countless pathways and crossroads and dead ends. And then, considering all this complexity, how is it possible for so many reactions to occur simultaneously, but in an orderly way inside each living cell? The orderly way is made possible by special protein molecules that you can see here, called enzymes. Each metabolic reaction requires its own enzyme to facilitate and control that specific reaction. And here you can see the enzyme involved in one of the previously analyzed pathways. Its name is aconitase, and you can see also the chemical reaction promoted by this enzyme, the conversion of citrate, a metabolic intermediate, into isocitrate. What I would like to emphasize here is the complex molecular structure of this enzyme. This whole constellation of atom groups is necessary to bind to the molecules and convert them. Enzymes are active inside the cells, whose internal structure is extremely complex and populated by many other proteins. And enzymes can carry out accurately targeted processes. In other words, they can recognize with high specificity the molecule they have to convert. So they are selective, and they know what to transform. Here you see how the citrate molecule is converted by the enzyme, and then another one, and so on and so forth. This very same process occurs at the same time in millions of enzyme molecules working simultaneously, each one controlling its own reaction. And this happens in each one of the 100,000 billion cells making up our body. In order to understand in a simple way how enzymes work, we can have a look at this cartoon. An enzyme makes possible a chemical reaction that otherwise, without the enzyme, would require too long because of the strong energy barrier. In other words, these special proteins open up smart shortcuts, allowing the reaction to occur smoothly. The enzymes control the access to the shortcuts by lifting or lowering its gateway. And this happens in a way that is strictly dependent on the whole context the enzymes are in. Therefore, there is an impressive enzymatic regulating system controlling all metabolic flows. We may think that understanding reproducing the chemistry of biological molecules would be enough to explain the basic functioning of the vast majority of living organisms. However, this vision would disregard a major chemical peculiarity of the molecules we are made of. The molecules synthesized by our metabolism have a very special feature. They are reconfigurable and can mix up in several ways or fold over themselves according to the presence of other molecules. This reconfiguration process is controlled by very weak forces which allow great flexibility. For instance, the change in the shape of the proteins located at the surface of cellular membrane allow to send signals inside the cell about what is happening outside. Or the membrane lipids can form detaching droplets. Or again, the DNA, when needed, can temporarily shrink into small aggregates, the chromosomes, in order to open up and stretch again at a later stage. These sort of collective molecular dense moves are crucial for life processes. what you said highlights a very important aspect. If we were to put together the huge set of metabolic molecules and enzymes, including thousands of proteins, millions of billions of molecules, well, even all that would not be enough to create a living organism. In order to have a living organism, we need to frame all that in a self-regulating confinement structure. Even in its smallest unit, this structure is amazingly complex. And this structure is nothing but the cell. The intricate web of metabolic reactions is essential for the survival, growth, and development of a living organism. However, what we define as living being cannot be reduced to a set of chemical reactions. Properly ordered metabolic processes need a specific organizational structure separate from its surrounding environment. The smallest version of this very crowded space is called cell. A cell could be compared to a small fortified city protected by a phospholipid protection wall, whose function is to strictly control the accesses and monitor what happens outside. The city is efficiently divided into compartments. Cells are inhabited not only by enzymes, but also by many other kinds of proteins carrying out the most diverse functions. Some of them are embedded in the cell membrane, others are in charge of the communication with other cells. Others are considered as sentinels, monitoring the outside and able to warn the cell if need be. Some of them play such an important role that every cell can display on its surface up to a couple of thousand of copies of a single protein type. However, cell complexity is not limited to its outside. For instance, if we investigate the inside of a human cell, we can notice large supporting structures made of other kinds of proteins. This is the cytoskeleton, a complex scaffolding system created according to the cell needs. When portions of such structures are not needed any longer, they are disassembled and their components are recycled. Everything is in progress and nothing is wasted. These are microtubules, rail-like ways connecting the different city districts, and used by other proteins to transport large vesicles from one cell side to the other. Even some organelles, like mitochondria, use them to move inside the cell. Cell complexity is as fascinating as mysteria. The immensity of cosmos and its ever-changing nature keep surprising us as much as the beauty of the cell microcosmos, which is an indispensable condition for any living organism. The most amazing feature of a cell is its intricate architecture that keeps self-building itself using energy and matter constantly converted through metabolism. Cells are very much like buildings, but at the same time they also resemble the continuous construction processes leading also, through cell division, to the creation of new standalone buildings as replicas of the original one. At the same time, the ability of cells to grow and self-adjust the shape of their own structure requires a system able to make new copies of its components, namely of its thousands of different protein types. Even if this may sound unsettling, we also observe that life is intrinsically fragile, and this is not due to some design mistake or to an unfortunate evolutionary outcome. Chemical fragility is essential for living systems. because it allows molecules to be degraded, to be deactivated, and also to be removed. Even the connection between molecules need to be somehow fragile in order to be able to change, to be able to develop and mature, starting from more elementary cells. Fragility is the high price paid by life in order to be dynamic and capable of undergoing evolution. Oddly enough, transience and mortality end up being key ingredients for the functioning of living organisms. What you said is really deep and true. We previously showed some diagrams illustrating the fundamental role played by proteins. Actually, there is another key factor that is essential for the processes we have seen so far relating to the growth and development of new cells. And this key factor is the ability to continuously synthesize new proteins. Proteins are also deciduous, and this intrinsic fragility is not accidental, but strictly related to the role they play. Many proteins are synthesized and produced only as response to specific perturbations. But once the perturbation is over, their levels have to go down again, otherwise they could become detrimental. Therefore, synthesis, degradation, and resynthesis of proteins are key life structural phases. Cyclins are proteins providing an extreme example of this kind. They regulate the cell cycle and, as shown on this chart, they pile up during the cell growth phase, between cell division phases, the so-called interphases. And then once cell division has been completed, they are quickly degraded. Therefore, there is a continuous process of synthesis and degradation, resynthesis and degradation, which is key for controlled cell division in an organism. Now, a key question emerges. How are proteins synthesized? And how do living systems self-build themselves? In other words, the answer to this question allows us to emphasize the role of two extraordinary features about which the world has not felt sufficiently surprised yet. The first one concerns the fact that each cell in any organism already contains some sort of coded instructions on how to synthesize dozens of thousands of different proteins. These instructions are provided by sequences of nucleotides, which are the building units of nucleic acids, and in particular of DNA, a molecule in which the instructions are stably deposited. In other words, thanks to these instructions and these sequences, it is possible to replicate identical formed copies and pass them on across generations of cells and organisms. The second extraordinary feature related to this one is the fact that DNA nucleotide sequences that can be copied into RNA molecules called messenger RNA, which can then be translated into amino acid sequences building up proteins according to a set of general rules known as the genetic code. Therefore, at the heart of protein synthesis and at the heart of any living organism, there are a code and a translation process. As a consequence, the resulting process is nothing but a purpose semiotic process. Each cell has an embedded internal design representation in the form of DNA filaments, thanks to which it can generate new copies of the required proteins. The necessary instructions are in the DNA under the form of sequences of 4 nucleotides. Each possible sequence of 3 out of 4 nucleotides corresponds to one of the 20 amino acids, the building blocks of proteins. However, because of the lack of chemical affinity and steric correlation between them, nucleotide triplets and amino acids would not be able to interact. The correlation between the two sequences is made possible by the genetic code that works like a dictionary, translating the DNA language into protein language. The translation process is possible only thanks to an adapter, that is, an entity interacting with both systems and filling the structural gaps as if it was a key able to open only its matching lock. This adapter picks up and puts together the unequivocally matching elements. This adapter is also what provides meaning to the initial sequence, because without a code, it would remain a simple set of similar elements, which would not differ from other similar ones. Once a code is available, not only a particular sequence becomes distinguishable from all the others according to its outcome, but the code is also the key condition to have an outcome. The pattern of this semiotic process, in which information and meaning play a key role, also represents what happens inside the cells. The gene that needs to be expressed is taken from the matching DNA portion and copied onto a messenger RNA filament. The mRNA filament released by the nucleus is then placed inside the ribosome, the cell factory, in order to let it interact correctly with special RNA molecules called transfer RNA or tRNA. These molecules are the core of the genetic code. They are the adapters matching nucleic acids with proteins. The structure contains on the one side an amino acid and on the other a nucleotide-based triplet, which is complementary to the mRNA sequence. When the tRNA enters the ribosome, it interacts with the filament. When the filament triplet and the adapter tRNA match, the amino acid linked to the tRNA is added to the protein chain as soon as it is assembled. Residual tRNA is released through the filament, sliding up to the next triplet, and then the whole process starts again. Thanks to the tRNA, the meaning of the information present on the messenger RNA is then decoded under the form of the amino acid sequence. The replicability of the process is guaranteed by the fact that the triplet amino acid pairs established by the adapters are fixed and identical in every living organism. However, it is important to note that there is no necessary correlation between the two sides. This is further proved by the fact that through biotechnological manipulations, it is possible to change the matches, triggering the biosynthesis of brand new proteins. We thus could say, using an expression coined by biologist Marcello Barbieri, that proteins are molecular artifacts. Their production cannot be uniquely understood through chemical reactions, but also through a semiotic process requiring information and meaning assignment through a code. The already extraordinary possibility to continuously produce proteins based on a code and a set of instructions has some key repercussions for all living organisms. The first one is the reproduction process. The fact that the DNA can generate two identical couples of the same molecule allows the reproduction of the instructions in their transmission to further generations. And this fact guarantees a continuity over time that has been going on for millions, billions of years. A second, more subtle, but equally important repercussion is the possibility of a targeted and differential use of these instructions. A clear example is the specialization of our tissues and our organs. Not all cell types carry out, and not all cell types are characterized by the same metabolic pathways. Some of these metabolic pathways are active in some cells, but not in others. This picture is taken from a very recent study in which the concentration levels of thousands of proteins in the main human organs and tissues was accurately measured. Each column that you can see corresponds to a different kind of tissue, and the different profiles of the colored bars show that in some cell types, some metabolic pathways out of those composing the metabolic network we have previously seen are particularly active, while other tissues feature other metabolic pathways. What does this mean? It means that the instructions, which are the same in all cells, are used differently. Some are executed and some not. The same phenomenon applies even if on a different scale on the different cell types populating a human body. Here you can see 6 different images of 6 different cells. They look very different from each other, highly specialized. However, all these cells have self-built themselves using the same set of instructions. There is clearly a different and highly sophisticated use of such instructions that opens up the huge scenario of gene regulation, or more generally of epigenetics, which defines this possibility organisms have to use the same instructions in highly different and sophisticated ways. We can conclude that cells and organisms are not the inevitable result of the instructions included in their DNA. Cells with the same instructions can be very different from each other. And that is exactly what epigenetics is about. Some say that the DNA proposes and the cell disposes. This could be an interesting point of view, but would still be reductive because no cell can dispose anything alone. Cells are always influenced by other cells, which on their turn are influenced by other cells, and all of them together are influenced at an upper level by the whole organism, which on its turn is influenced by other surrounding organisms and also by the surrounding non-living environment. Essentially, in order to keep becoming what they are, cells and organisms cannot separate from what is other than them. This reflection introduces the main topic of the next leg of our journey, which will be entitled Being Alive, the Self and the World. After that, there will be a third leg during which we will see how this huge potential plasticity of living organisms that we might expect leading to some unregulated complexity quite surprisingly includes some general, simple, and universal rules. And for instance, we can consider that the average life of vertebrates depends on their average size. And this very same rule applies to all of them. So the same rule applies, for instance, from mice to whales. During the fourth leg, another topic will be dealt with, namely how plasticity of living organisms led to that extraordinary mechanism called evolution, thanks to which, in the course of history, events developed so many different shapes and how all that was possible. All that said, we can recognize the fact that interest for animals and for living beings that began such a long time ago, as documented by the cave paintings, led us to discover and gather a great deal of knowledge that would be absolutely unimaginable for the men living in the caves of Chauvet. But are we really so different today from those men of 32,000 years ago? Since then, incredible scenarios have kept opening up, unveiling exciting new possibilities of the cellular and molecular worlds. At the same time, discovering that this familiar condition of being alive depends on so many strange and countless factors is somehow equally unsettling. Certainly, we are excited and sometimes puzzled by new findings, but maybe we still hold the very same sparkle that motivated those faraway artists, the ones who made the cave paintings.